A rare genetic mutation has made some individuals remarkably resilient to viral infections, even against pathogens that would typically incapacitate most people. This biological phenomenon was first observed in a small group of individuals, over a dozen people scattered across the globe, who exhibited an extraordinary ability to resist diseases like the flu, measles, chickenpox, and even a sneeze. While their bodies struggled with certain bacterial infections, viruses could not establish a foothold. This resistance was initially noted by Dr. Dusan Bogunovic, an immunologist at Columbia University, who investigated a genetic mutation associated with increased vulnerability to bacterial infections 15 years ago. He stumbled upon an unexpected connection, a deficiency in a molecule called ISG15, which acts as an immune regulator. What intrigued him most was the type of inflammation these individuals carried, a form of antiviral immunity that surprised him. "That's when I realized they were hiding something," he recalls. Further investigation revealed that immune cells had encountered viruses such as the flu, measles, mumps, and chickenpox, yet none of the patients showed symptoms. "If we could produce this kind of light immune activation in others," he says, "we might protect them from almost any virus." Now, this concept has become reality. In a recent study, Bogunovic's team demonstrated an experimental therapy that temporarily restores the same antiviral immunity as those with the ISG15 deficiency. The treatment involved administering a nasal spray containing 10 strands of messenger RNA, each enclosed in lipid nanoparticles. This formulation blocked the replication of influenza and SARS-CoV-2, while significantly reducing disease severity. Instead of directly suppressing ISG15, the therapy activated 10 key proteins from the body's 60 normally produced by ISG15 deficiency. These proteins play a critical role in broad-spectrum antiviral protection. The delivery system draws inspiration from current COVID-19 mRNA vaccines. Once inside the cells, the mRNAs guide the production of protective proteins. We only generate a small amount of these 10 proteins for a very short time, explains Bogunovic. This leads to less inflammation than seen in ISG15 deficient individuals, but the inflammation is sufficient to prevent viral diseases. The team envisions their technology as a rapid response shield for future pandemics. It could protect frontline workers, vulnerable populations, and exposed family members even before the virus is identified. Crucially, the therapy does not interfere with the body's ability to build long-term immunity. However, challenges remain. The delivery system needs refinement. When administered to animals via nanoparticles, Bogunovic notes, the 10 proteins were produced in the lungs, but probably not at high enough levels to make us comfortable going into people immediately. Once the therapy reaches our cells, it works, but the delivery of any nucleic acid, DNA or RNA into the part of the body you want to protect is currently the biggest challenge. The study was published in Science Translational Medicine, https://doi.org/10.1126 citransmed.adx 5758.